Great. So if we're going to stay on schedule, yep, Sara is next. Sara Wilmsen is the last speaker of the session, and the title of her talk is The Effect of Acclimation to a Fluctuating Temperature on CO2 Production During and After Exercise in the Desert Tarantula Grammastola rosea. So Sara, do you want to go forward?
Thank you. Yeah, so this was a project that I did with some undergrads during a class that we have here at my university. So they used some spiders that I had in the lab. So temperature is really important for ectotherms. As many of you probably know, temperature affects a lot of aspects of physiology, including enzyme kinetics. So at higher temperatures, you have greater reactivity of enzymes and things like that. It also affects membrane fluidity, which can affect how things are going across membranes in the cells. And of course, it affects metabolic rate, which ties back into the enzyme kinetics as well. And all of these things can impact size. If you have a lower metabolic rate, you're probably not going to grow as fast. It can impact reproductive capacity, and it can impact how much food you need to maintain your lifestyle. So there's this idea of metabolic compensation, which is even ectotherms which don't maintain the same temperature can, though, acclimate to a specific temperature over a given period of time. So the standard metabolic rate in ectotherms can be changed to increase fitness at any specific temperature. And this can be done through enzyme isomers, so producing different enzymes with slightly different conformations, and also through behavioral thermoregulation. And so this is a graph that shows that concept where you have an organism acclimated at 15 on the blue line, and an organism acclimated at 30 degrees in the orange line. And so you can see at 15 degrees and at 30 degrees, this low temperature acclimated species organism has a higher metabolic rate at both temperatures, and than the 30 degree acclimated animal. So in the laboratory, a lot of times when we're looking at studies focused on temperature, we use a constant temperature because it's easy. We can just set a incubator at a specific temperature and leave it, right? So usually either a high or low temperature or both. But for most all organisms, this isn't relevant for their actual ecology, right? These are some graphs that I found of temperature in Douglas fir forests and montane forests, and this also gets into like microclimate as well. So even though there's changes in temperature, or maybe there aren't as big changes in temperature in the overall environment, they can be different in the microclimates as well. But the main takeaway is that organisms don't generally live at a constant temperature. This is especially true for our study species. We used Grammastola rosea. They're a desert tarantula species. So in deserts, of course, the temperature fluctuates a lot. This is a graph of temperature, daily temperature in the Atacama Desert, I believe. And the maximums are up around 30, and the minimums are down below 10 degrees C. So that's a huge range in temperature that they're having to deal with. So, and because they are fairly small for an organism, they're very temperature sensitive, and they're ectothermic, so they're ambient temperature dependent mostly, besides behavioral thermoregulation, sorry. And yeah, so there's a lot of environmental fluctuations there. So the aim of this study was, what are the effects of acclimation to a fluctuating temperature, which is what they normally see in their environment. And we wanted to look at standard metabolic rate, which is a resting metabolic rate at a specific temperature, and then also during and after exercise. So what we did is we got captive bred spiders, We acclimated those spiders at a high temperature of 30 degrees C, a low temperature of 15 degrees C, and then a fluctuating temperature that fluctuated every 12 hours, along with their light cycle, and it changed between these two temperatures. So at night, it was at 15 degrees, and during the day, it was at 30 degrees. And this is a graph of actual measurements from our incubators. We used flow-through respirometry to measure routine CO2 production at 2 different temperatures. So these are our acute temperatures or run temperatures in the graphs that I'll show you later. So we ran them at 30 degrees and we ran them at 15 degrees. And we had 2 different flow rates to adjust for the differences in the metabolic rate that we were expecting to see. So we run these guys on a treadmill. We built a treadmill that we could measure metabolic rate on. Here's a little video of one of them running on the treadmill. We ran them for 5 minutes at about 100 centimeters per minute. And then we measured their oxygen or their CO2 production for a half hour post-exercise to get an idea of how much oxygen debt they're accumulating during their run. And that's called the excess post-exercise oxygen consumption, or EPOC. For our statistics, we used an ANCOVA to take out the effect of carapace length and mass. These are the masses for each temperature acclimation and the carapace lengths as well. We estimated the marginal means of each of these, both standard metabolic rate and CO2 production during exercise. And then we used an effect size, which is Cohen's d, to look at the effect of treatment. So this is kind of an maybe confusing slide, so I'm going to go through this, but the other 2 slides are exactly the same. So on the left over here, we have CO2 production in microliters per hour, and then we have our 2 run temperatures, and then in the blue, we have our animals that were acclimated to 15 degrees. In the red, we have our 30-degree animals, and in the green, we have our fluctuating acclimated animals. These are our Cohen's d values. So these are effect sizes for pairwise interactions or effects between this. The red one is 30 compared to 15. The green one is fluctuating compared to 30. And then the blue one is 15 compared to fluctuating. And these 90% confidence intervals, if they overlap with 0, that means there's no real effect that we see. So in this graph, what this is showing us is that, like we expect, the 30-degree acclimated animals and the 15-degree acclimated animals actually did differ in their metabolic rate. And you can see that here and here at both temperatures. And then the fluctuating and the 30-degree animals did differ as well. There was an effect of that acclimation. However, the 15-degree animals and the fluctuating animals did not have an effect. We didn't see an effect of that. And I'll talk about maybe why we see those that later. For CO2 production during exercise, this was the 5 minutes that we ran them. We just took the total CO2 production. And so you can see that here. Again, these slides are very similar to the other ones that I showed you already. So here we have our pairwise Cohen's d values. And as you can see, we didn't see any effect of CO2 production during exercise between any of the groups. The EPOC was that half an hour after each run. We measured total CO2 production during that interval. And these were our results for that at 15, 30, and fluctuating acclimation. And we didn't see much effect here, except the 15 and fluctuating at 30 degrees at the higher temperature. We did see an effect of where the fluctuating was actually had higher CO2 production than the 15-degree acclimated animals. So spiders showed metabolic compensation like we expected. So when they're acclimated to a constant temperature, they do change their physiology in order to maximize the fitness of their metabolic rate. Was interesting was that the standard metabolic rate of fluctuating acclimated spiders was not different from the standard metabolic rate of the 15-degree acclimated spiders. And so that actually results in a higher metabolic rate at any given temperature during the day for these fluctuated animals, which is interesting and has interesting implications of their ecology. Like, is it more advantageous for them to have higher metabolic rates or to have lower metabolic rates? And I think probably it would depend on the sex. We didn't have these guys sexed out, so that could be an additional study that we could do. But acclimation did not produce a predictable effect on CO2 production during exercise or EPOC. And that could be because we could have had a bigger sample size, or maybe we would see some differences. It's an interesting thing, but I think really is what it is they're just very limited in how much they're in their aerobic capacity, just in general. So for future projects, I'd like to know, why does the fluctuating act more similar to our lower acclimated than our higher acclimated spiders? Because they were seeing both temperatures. And so I want to look at the artifact that may be caused by timing, because we ran these guys during the day, and that was during their 30-degree period, but it was right after their 15-degree period. So I wonder if we ran them at night, we would see an opposite effect, maybe. Yeah, and then just talking about, like, is it more advantageous for them to have a higher metabolic rate or a lower metabolic rate? And then just having a larger, more consistent sample size, I think would be good. I think next time I would like to get actual live caught or wild caught organisms that would be more indicative of the actual ecology of these organisms. So I want to acknowledge my 3 undergrads that I did this project with, Daniela, Stephanie, and Al. They were all really interested in this project and really did really good work. I want to thank my advisor and the Jalowski Lab for helping us get through this. The Howard Hughes Medical Institute grant, which funded this project, and the University of North Texas. And if you saw my last talk, you know what's coming. I am seeking a postdoc position for fall 2022. I'm hoping to finish on my dissertation next spring. This is my email address. If you have a project in mind, or you want to develop a project together, or you're looking for a postdoc, just let me know, or you know somebody that is looking for a postdoc. I'm interested in the ecophysiology of arachnids, how these, especially climate change, how it affects these spiders. And I mean, after seeing all these scorpion talks, I'm open to anything. Also, respiratory morphology. If you saw my other talk, that was what it was about. I'm really interested in how respiratory morphology changes between spider species. I think it's so fascinating, the different tracheal morphologies that occur in these spiders. And I'm also interested in the effects of toxins on spider populations as well. And so, yeah, I know I'm a little early, but I would love to take any questions that you have.
Thanks, Sarah. Great talk. So we have maybe 2 minutes for questions if we're sticking on time, and there are several questions. So just going back here, I see Cara Shillington says, how did you ensure aerobic activity on the treadmill?
We didn't. So we assumed that these spiders were going to be anaerobically producing or anaerobically exercising. And that's why we went ahead and looked at the post exercise oxygen consumption, or production,
Okay, Louis Roque says, what effect, if any, do you think altitude would play in this data? Any plans to replicate the study utilizing differences in altitudes and pressure into account?
Yeah, I mean, that could, altitude definitely could have an effect. I mean, if the pressure is different, that could influence how well they're getting oxygen to their tissues because of their book lungs. yeah, I don't have any plans for it. This was an undergrad project, but I'm definitely interested in that if someone wants to take me down to Chile.
Okay, so maybe one more question. So Jillian Cowles says, are these tarantulas known to be completely ectothermic? Any endothermy could affect the acclimation.
As far as I know, there is no endothermic capacity of these spiders. I do work with endothermic insects in my PhD, and their metabolic rates are very high compared to these tarantulas, especially per gram. So I wouldn't expect there to be any endothermy. They also don't have a lot of spiders in general. Just don't, in from what I know, don't have the capacity in their musculature for endothermy, and I can't think of a good reason for them to expend that much energy, especially when you think about their lifestyle, which in these tarantulas would be mostly sit and wait, at least for the females, and mostly sit and wait for the males as well, except when they're seeking mates. So yeah, not that I know of.
Great. So maybe in the spirit of sticking to the schedule, thanks again, Sarah. And I think there may be one or two more questions and lots of kudos in the chat window that you can address and check out. And I'm sure the organizers have announcements to make.